Myself, Dr. Vijay Anand, Associate Professor, Department of Life Sciences. Today's topic is glycolysis. So, glycolysis is a major pathway for oxidation of glucose. So, where this glucose is oxidized into 3-carbon compound, especially, so that is pyruvate. And this glycolysis includes a series of enzymatic reactions that takes place in the cytosol. So the entire glycolytic reactions are divided into 2 phases. So the first phase we consider as preparatory phase, and the second phase we consider as payoff phase. So preparatory phase we consider as energy consuming reaction, whereas payoff phase is considered as energy yielding reaction. So when you consider the reactions under the preparatory phase, so glucose is a starting substrate, and glucose is phosphorylated in the first reaction with the presence of ATP, and the enzyme catalyzed this particular reaction is glucokinase. So hexokinase is a non-specific enzyme, whereas glucokinase is a specific enzyme, so which catalyze this particular reaction where glucose is phosphorylated to glucose 6-phosphate. So after forming glucose 6-phosphate, so this is a hexo sugar, where this hexo sugar is converted to keto sugar. So again glucose 6-phosphate to fructose 6-phosphate conversion will take place. So this reaction is mainly catalyzed by phosphohexoisomerase. Then fructose 6-phosphate, again it undergoes phosphorylation where ATP is involved in this reaction. So this reaction is catalyzed by the another enzyme, that is fructokinase. So for all these phosphorylation reactions, mainly kinase or the group of enzymes which are involved, which requires ATP as for the phosphorylation. So when fructose 6-phosphate gets phosphorylated, so it forms fructose 1,6 biphosphate. After forming fructose 1,6 biphosphate, so this is cleaved by this specific enzyme called aldolase. So when it cleaves fructose 1,6 biphosphate, it forms glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So these two are readily interconvertible compounds. So glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate, and similarly dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate. So this interconversion is mainly enhanced by triose phosphate isomerase. So after forming glyceraldehyde 3-phosphate, so the further reactions, so that comes under payoff phase. Payoff phase is mainly energy yielding reactions. So where the glyceraldehyde 3-phosphate so is mainly converted to 1,3-bisphosphoglycerate. So here, so glyceraldehyde 3-phosphate so mainly utilizes NAD for the reaction, and this reaction is mainly catalyzed by glyceraldehyde 3-phosphate dehydrogenase. And this is a first energy yielding reaction where 6 molecules of ATPs were formed from this particular reaction. After forming the 1,3-bisphosphoglycerate, so again this bisphosphoglycerate is converted to 3-phosphoglycerate. So this reaction is particularly catalyzed by phosphoglycerate kinase. And the 3-phosphoglycerate, so further it is catalyzed by phosphoglycerate mutase. So where the phosphate which is present in the 3rd position is shifted to the 2nd position, which is catalyzed by phosphoglycerate mutase. So this is not an energy yielding reaction. So after forming 2 phosphoglycerates, so there are 2 more reactions that leads to form pyruvate. So 2-phosphoglycerate is mainly catalyzed by enolase, so that it forms phosphoenol pyruvate. And after forming phosphoenol pyruvate, the final reaction, so it is catalyzed by pyruvate kinase. So this is again the 3rd energy yielding reaction of glycolysis. So again the phosphoenol pyruvate is converted to pyruvate. So all these reactions which comes under the energy yielding reaction, that is payoff phase. So coming to the energetics of glycolysis, so the energetics of glycolysis can be discussed under 2 broad categories. We can consider as energy yielding reaction as well as energy consuming reaction. When you talk about energy consuming reactions, so that comes under the preparatory phase, so where ATP is utilized for the phosphorylation of glucose as well as phosphorylation of fructose. When you talk about the energy yielding reaction of glycolysis, there are three important energy yielding reaction of glycolysis. One is the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, where it yields 6 molecules of ATP. Whereas the second energy yielding reaction of glycolysis is mainly conversion of phosphoenol pyruvate to pyruvate. And the third energy yielding reaction of glycolysis, we can consider conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. And when you talk about the net gain of ATP which is formed in the glycolysis is, so it produces 10 molecules of ATPs, but out of 10 molecules of ATPs, 2 molecules of ATPs have been utilized for the preparation of especially the energy consuming process. So the net gain is 8 molecules of ATP. So the energetics of the glycolysis can be calculated mainly based on the number of ATPs produced minus number of ATPs consumed. So the net gain of ATP is 8 molecules of ATP. And the important key enzymes involved in glycolysis, there are 3 important key enzymes which are involved in glycolysis, which includes hexokinase, phosphofructokinase and pyruvate kinase. And these enzymes, wherever these enzymes catalyzing this reaction, we consider as rate limiting reactions. So those rate limiting reaction, it includes conversion of glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6 biphosphate. Then third one is phosphoenol pyruvate to pyruvate. And these reactions, so there are irreversible reactions that takes place in the glycolysis. So those irreversible reactions of glycolysis are considered to be the basic reactions which are involved as substrates for the gluconeogenesis. And when you talk about inhibitors of glycolysis, so mainly the chemical inhibitors plays a very important role, which includes iodoacetate and arsenite are considered to be the major inhibitors. Talking about significance of glycolysis, so where this glycolysis takes place in our body, so glycolysis takes place almost in all the tissues, and mainly this pathway is meant for providing energy in the form of ATP. So, and there are various intermediates which are produced during this metabolic pathway, so which are connected with the oxidation of glucose. And this glycolysis is considered to be the direct oxidative pathway for glucose. And in the absence of oxygen, so here the glucose is oxidized, it is converted to pyruvate and then it is reduced to lactate. So this condition we call it as anaerobic glycolysis, and during this anaerobic glycolysis the formation of lactate is getting accumulated especially in the muscles, and which leads to form muscle cramps. And glycolysis is mainly under the control, so it depends upon the concentration of the hepatic glucose, that means level. So if excess glucose is there, so that means when it undergoes oxidation, so it increases the formation of pyruvate, and ultimately so it contributes to hyperglycemia, especially that leads to diabetes. Thank you.